We don't really hear you well. Would you please somehow make it a little bit louder?
Oh, okay. I can't hear you. And my question is just about the clinical application of the results for Dr. Zhong. Yeah.
I see. Okay. Yeah. Okay. So Dr. Zhong, if you're done with slides testing, could you please stop screen sharing so we can
Sure.
start off with our event background, our posters. Thank you so much. So you want to share this? Okay. Share it. Share screen. Okay, perfect. Thank you. All right.对，这样比较好。OK， 什么？OK， 谁？没关系，可以但是没有看，就是在可以看得到下面。也不放这个人是吗？跟谁？放一遍是可以的。
创办一所梦想中的大学，对得起中国灿烂的文化、悠久的历史，能够为世界文明、人类未来做出中国人应有的贡献。我们希望，我们就是这样一批走路的人、创路的人、筑路的人。西湖大学是一所社会力量举办、国家重点支持的新型高等学校。它的新体现在3个方面，那就是小而精、高起点、研究型。
根据国家法律和西湖大学章程，西湖大学实行校董会领导下的校长负责制，给校长最大的治校空间，给教师充分的治学自由。
新发现和新技术推动着社会进步，中国的发展需要顶尖创新人才，西湖大学就是要成为吸引和培养创新人才的基地。我们有采取国际通行的同行评议模式，学术成果就是唯一的评价标准，讲席教授都是各自领域的开创者或领导者，青年教授也是同龄人中的佼佼者。
I'm of course excited to see Westlake University, a unique place. I received many offers from different places to move from my previous university, but I decided to come here for the quality, for the prestige, and for the science. Talent coming from around the world to work together here on Westlake University to build this wonder research center and scientific center.
西湖大学正在建设多个科研平台与设施，努力为每一位科研人员提供最先进的服务。
开始吧。提醒大家 We have a minute countdown. We'll start sharp in about 30 seconds. Okay. Okay, okay. Welcome to the 5th webinar of Westlake Proteomic series, Proteomics and Cancer Research. It is hosted jointly by Westlake University, Westlake Laboratory, Guomix, iMarker Lab, HuB 2.0, PDPM, and Westlake Omics. We would like to pay a special tribute to Chinese Ministry of Science and Technology Belt and Road Initiative Innovative International Talent Program. We're honored today to have invited 5 distinguished speakers. Our speaker today is Dr. Qing Zhong, Group Leader of Cancer Data Science at Children's Medical Research Institute at the University of Sydney. We also have 4 panelists, Dr. Tak Yo Lo, an Associate Professor from UKM Medical Molecular Biology Institute at National University of Malaysia. We have Dr. Wen Guangshao, an Associate Professor from Shanghai Jiao Tong University. Dr. Liu Jiatian, a PhD student from Westlake University, and Dr. Tianan Guo, an assistant professor at Westlake University. Dr. Zhong is a data scientist with background in computer science, applied statistics, and cancer biology. He completed his undergrad study in computer science and obtained a PhD degree in biochemistry and computer science at Swiss Federal Institute of Technology Zurich in Switzerland. Since 2017, he has been the group lead of Cancer Data Science and Children's Medical Research Institute, and subsequently senior lecturer at the University of Sydney. His group aims to locate and mine big biomedical data to achieve the goals of personalized medicine with the focus of developing data-driven computational tools and sophisticated machine learning algorithms to predict the most effective cancer treatments for individual patients. Without further ado, let's welcome Dr. Zhong to share his talk today. Dr. Zhong, you probably need to unmute first.
Oh, thank you. Thanks for the introduction. Sorry, I was muted. Can you see my slide?
Yes.
Great. So let's get started. Thanks, Tianan, and thanks for the introduction and the invitation. It's my great pleasure to speak here today. My name is Qing Zhong, and I'm leading the Cancer Data Science team in ProCan at the Children's Medical Research Institute in Sydney. So before my talk, I will give you a very short introduction to ProCan, which stands for the International Center for the Proteome of Human Cancer. ProCan was seed funded by the Australian Cancer Research Foundation and supported by a range of national and international funding bodies. ProCan is regarded as a flagship program at the University of Sydney, which aims to create a global public knowledge base of cancer proteome from tens of thousands of cancer samples. So we hope that this knowledge base will help doctors to make informed treatment decisions for patients within a clinically relevant time frame, let's say 72 hours from sample acquisition to report generation. We also hope that it will help scientists to better understand cancer biology so that we might identify possible new cancer treatments. The genesis of ProCan can be partly attributed to a landmark paper published by Tianan and Rudy in Nature Medicine back in 2015. The paper described a technology that allows conversion of clinical biopsy samples into permanent digital proteome maps. This is certainly an enabling technology because in contrast to the current pathology assessment, where only a few proteins can be stained and analyzed at a time, this new technology enables high throughput quantitation of thousands of proteins in a single run for a very tiny biological sample. So here we can see some key numbers at the bottom of this slide. These numbers are our projection about how many proteomic maps or files can we generate in the next few years. So as you can see, we anticipate that using a variant of the this technology, PCT source, we will have reached a total of 100,000 proteomic files by 2026. So today I'll be taking you through a study in ProCan where we generated a pan-cancer proteome map of 949 human cell lines. We know that cell lines have been good models for our understanding of cellular process. And the molecular drivers of carcinogenesis and cell line models are also very good for identifying cancer cell vulnerability to both genetic and pharmacological perturbations. However, published work shown here on proteomic quantifications for cancer cell lines are either limited in the range of cancer types or the number of samples analyzed. Can you see my screen actually?
Yes, we can see. Yeah.
It's the correct mode, right?
Yes,
very well.
Yes, it is.
Very clear. Yeah.
Okay. So today I'll be So for these reasons, we started this cell line project collaborating with the Sanger Institute. So the initial questions we wanted to ask in the original project proposals were very simple. The first question we wanted to ask is, can we generate large-scale proteome measurements of the cell lines based on which other types of molecular and proteome phenotypic data have already been produced? And the second question was, can we get insights into biology beyond the genome and the transcriptome where we have the additional proteomic data? Moreover, we also wanted to know whether we can obtain new knowledge about cell type specificity, post-transcriptional modification, or protein-protein interactions. To answer these questions, we developed a deep learning-based pipeline that incorporates multi-omics, drug response, and CRISPR-Cas9 gene essentiality screens. We showed that this AI-powered pipeline reviewed thousands of protein-specific biomarkers, and it can predict cancer vulnerabilities with high performance. This project was being driven by Emmanuel from the Sanger Institute and Rebecca and Simon from my group. We have received the 949 human cancer cell lines from the Sanger Institute and got them proteomed in ProCan. We quantified the proteomes of these cell lines covering about 28 tissue and over 40 genetically and histologically diverse cancer types. So altogether, we have quantified almost 8,500 proteins. So on the right-hand side, we can see a radial bar chart showing the proportion of different tissue types. Each bar denotes a cell line, and the height of the bar indicates the correlation of the replicates. The bar chart shows high correlations between replicates of each cell line with a sample-wise median Pearson correlation coefficient of 0.92. So more specifically, let's take a look at figure 1. That is, each cell line was acquired with 6 replicates. So we had 3 sets of technical duplicates run by data independent acquisition on 6 of our mass spectrometers. Altogether, we obtained almost 7,000 runs with over 10,000 hours of mass spectrometry time. Using a data processing software called DYNE, we generated the largest pan-cancer cell line proteome map to date, significantly expanding previous efforts such as the Broad Institute's CCLE dataset. In other words, the contribution of our study is the addition of proteomic measurements to the existing independent molecular and phenotypic datasets, which spans more than 1,300 cancer cell lines as part of the Cell Model Passports and database, which is curated and managed by the Sanger Institute. Another contribution is that the drug response dataset was expanded by the Sanger team, as shown by the orange bars in figure C, highlighting the additional number of unique drugs presented in this study compared to previous ones. We then also looked at the correlation of the proteomes of each cancer type, tissue type, batch, and instrument. And then they were benchmarked with a baseline measuring the correlation between random unmatched sets of replicates. The results suggest that our dataset has a very high technical quality. And it is worth mentioning that the sample reception, data generation, and computational analysis of this huge dataset have spanned years and across multiple mass spectrometers. This is evidenced by this figure where we can see the timeline of data acquisition with colors denoting the batches and rows indicating the 6 mass spectrometers. With such a complex setting, we expect that batch effects were inevitable, and despite a very well-designed study. So to remove these batch effects and get reproducible results, we used both Dyn internal normalization method and software package called Pronom, where Pronom is a computational method that we developed in-house, which ensures that any amount of cancer samples can be quantitatively compared, regardless when these samples were processed and on a very large scale. So after data pre-processing and batch correction, a way to look at the data in its entirety is to show it as a heat map. Here we have row-wise sorted the cell lines according to tissue types, and column-wise we have sorted protein intensities. You can see there is a set of proteins that are uniformly and detected across cell lines, and these will be our housekeeping proteins. There's also a set of proteins that are less uniformly presented, but show clustering according to particular tissue types. And these will be our tissue-specific proteins. So obviously, it would be challenging to identify interesting patterns in a heat map with thousands of proteins. So we have used a dimensionality reduction method, UMAP, that transforms the data from high dimension into two-dimensional subspace. And let's take a look at figure A, which shows that UMAP indeed reveals some grouping by tissue of origin, such as the lung and skin cancers. We can also see that blood and lymphoid cells show distinct molecular profiles, which corresponds to the published CCELE dataset. So we were very happy about this external validation, although it's very simple. Within the blood and lymphoid tissue types, we can further observe subclustering according to their cell lineage. These results tell us that our quantitative data are able to accurately map out known biology, underlining these cell lines, particularly according to tissue types and cell lineage. This is a very busy slide showing a variety of computational analyses, including MOFA. So due to the scope of my talk today, I would like only give you 3 takeaway messages. So first is there's only moderate correlation between transcript and protein abundance, which is consistent with the effects of post-transcriptional regulation. Second, our dataset and study capture additional protein-specific information that can augment our understanding of genomic alterations. Third, known and unknown protein-protein interactions and co-regulation are better captured by proteomics rather than transcriptomics and CRISPR-Cas9. So together with the fact that we identified more than 1,000 putative protein-protein interactions that are not reported in any of the protein network resources, these results actually tell us or may tell us that we can use pairwise protein correlations to identify novel putative protein-protein interactions in future work. So we then also considered how to use proteomic data to identify biomarkers predictive of cancer cell line response to drug or CRISPR-Cas9. So for this, we expanded the previous Sanger pharmacological screens to include a total of more than 570,000 IC50 values, which measure how much concentration is needed to kill half of the cells. So altogether, we have a total of 625 unique anti-cancer drugs, which is about 48 increase in unique drugs over previously published datasets, including FDA-approved drugs, drugs in clinical development, and investigational compounds. For CRISPR-Cas9, we had genome-wide essentiality measurements for 17 6,486 genes. So we identified many known associations as shown in these two figures. Let's just take a look at the left figure for drug and protein associations, where each association is represented by the linear regression effect size and its statistical significance on the X and the Y axes. The color is coded by the distance between the target and the associated protein in a string network. The letter T denotes the associated protein is either a canonical target of the drug or CRISPR-Cas9. And the numbers represent the minimal number of interactions separating the drug or CRISPR-Cas9 targets to the associated proteins. And finally, the symbol minus denotes an association for which no path was found. So we could see that among the strongest significant associations, 57 drugs were associated with the protein abundance of their canonical targets. And most drug responses that are associated with the target protein are negative associations. And for another 132 drugs, we could identify significant associations with proteins functionally related with their targets, that is just one step away in the string network. Finally, we can also see the many predicted protein associations are not with the target shown as blue dots. So next, we try to investigate if we can discover novel biomarker associations that are unique to the proteome and could not be predicted by gene expression alone. So let's take a look at drug response prediction first. With 949 cell lines and 625 drugs, a possible outcome is that we may find new scenarios where the use case of existing drugs can be extended. As for discovering new drug targets with CRISPR-Cas9, the idea here is that inhibiting certain proteins could selectively kill cancer cells. And for both tasks, we wanted to ensure the following three conditions during computational modeling. First is drug response for CRISPR-Cas9 should be well predicted by machine learning. Second, a drug should kill only tumor cells while keeping normal cells unaffected. Last, identifies simple explainable biomarkers so that they are applicable in clinics. So to satisfy these three conditions simultaneously, we developed a workflow called DeepProme, which stands for Deep Proteomic Marker. It integrates protein-wide association analysis with a deep neural network architecture called DeepOmicNet. So DeepProme can prioritize drug response or CRISPR-Cas9 gene essentiality that are highly predictive and specific to cancer cell lines using two selection criteria, namely predictive power of the DeepOmicNet and the selectivity of drug response or CRISPR-Cas9. By doing so, we were able to get a list of putative biomarkers for further downstream analyses. To see DeepOmicNet mathematically, we can formulate the Tolbert problem as a regression. That is, we were looking for a relationship between the protein matrix and the 625 IC50 values for drug response. And the relationship is modeled using a nonlinear function whose unknown model parameters can be estimated from data. Similarly, we were looking for a nonlinear function for CRISPR-Cas9. Here we used deep learning to model this nonlinear function. As you can see, a typical deep learning model, as shown here, is implemented by a multi-layer perceptron where all layers are fully connected. By contrast, our model is based on skip connections and grouped bottlenecks. And here, skip connections enables deeper and wider networks that can model complex interactions as our case. And to make these interactions more efficient, neurons within a layer are broken into groups to form bottlenecks, thus reducing the number of parameters. One major benefit of this architecture is that only one model needs to be trained for all drugs, which is easy to scale up to thousands of drugs in our study. So however, traditional machine learning methods need to train one model for each drug at a time. We also investigated the predictive power of deep omic net by using Pearson correlation as the evaluation metric. Here, as you can see, that deep omic net consistently outperformed elastic net and random forest across all omic data sets we used in our study. Previously, we defined the predictive power as one of the two selection criteria for candidate protein biomarkers. The second one is the selectivity of a drug or CRISPR, which is measured by a Fisher-Pearson coefficient of skewness. Here, the rationale is that a large skewness has a long and symmetrical distribution, as shown here in the figure for the drug linipanib. This means only a small fraction of cell lines are sensitive to the drug and thus have less side effect, which is needed. So now with predictive power and selectivity as the two selection criteria, we arrived at a set of candidates, which are 67 drug responses and 62 gene essentialities, with a total of 7,698 drug protein and 5,823 CRISPR-Cas9 protein associations. We then used the same linear regression strategy to highlight markers that are only evident at the proteomic level. We could see that predictions were significantly improved when compared to models that considered gene expression measurements alone. Here is just a zoom-in version of these 67 drug responses and 62 CRISPR gene essentialities. When we looked at the tissue-specific CRISPR-Cas9 response and identified more than 1,500 tissue type level CRISPR and Cas9 protein associations. So among the strongest was the dependency on FOXA1 transcription factor knockout and protein levels of BSG. And BSG is a plasma membrane protein expressed in breast cancer cells. Interesting part of this association is that this association was not observed at the gene expression level. And the FOXA1 BSG association occurred in luminal A and B and HER2 positive non-basal breast cancer cell lines, in which BSG protein abundance is low relatively to basal cell lines, as shown here on the left figure. And the BSG has been shown implicated in breast cancer progression and is a marker of the aggressive subtypes. And it was also shown to be associated with poor overall survival. So taken together, our result suggests that BSG protein expression is associated with basal-like breast cancer cells, whereas luminal and HER2 positive breast cancer cells with low BSG expression have an increased dependency on estrogen receptor-driven FOXA1 transcriptional activity. We also benchmarked our data set with others to understand whether the results are comparable. The independent data set we used is from CCLE. We could observe that the two data sets had comparable performance when predicting 3 external drug response data sets, as well as CRISPR-Cas9. We then investigated and found out that the predictive performance between transcriptomic and proteomic data were highly consistent for both drug response and CRISPR-Cas9 predictions. The predicted performance of CRISPR-Cas9 was generally lower because we think the knockout of most genes does not lead to lethality. With this result, the clinical implication is that using a protein assay for cancer treatment decisions might be a better option. Biologically, we have the potential to explore novel predictions and pathways made clear only through proteomics. One of the last analyses we wanted to perform is to determine how drug response prediction was influenced by the number of proteins. I think it's a very valid question because with 8,500 people, especially computational scientists, they may want to know, how many proteins is needed to have a very similar predictive performance. So for that, we performed or designed a randomized algorithm with a decrease of 500 proteins in each step. We find out that a subset of only 1,500 proteins was already able to provide 88% of the predicted performance of the full data set. To further explore the relationship between predicted performance and the subnetworks comprising proteins of different frequencies or sample support, we defined 3 protein categories with varying degrees of sample frequencies. So category A is defined for proteins that can be found in 90% or more cell lines. And category B is something between 20% to 90%. And category C is for less than 20%. So using a similar random downsampling strategy, but with a stepwise decrease of 250 proteins, we can see that proteins with higher sample frequency or sample support, which is category A proteins, provided the highest predicted performance as expected. In terms of network analysis, proteins with higher sample frequency had a significantly higher degree of connectivity in the string protein network. So collectively, these results demonstrate that a small subset of commonly expressed proteins with highly interconnected networks can already predict cellular phenotypes with high confidence. A related concept in genomics is called a network pleiotropy model. You might be interested to find that out. So this is my last slide, where I have summarized our study with 4 key points. Instead of re-emphasizing these points, I'd like to state the alternative. So this slide, this data set is now publicly available and represents a major resource for the scientific community and for biomarker discovery, and for the study of fundamental aspects of protein regulation. So I think it can be also used as a validation data set for cancer tissue cohorts and remined countlessly for any technical purposes by any computational scientists. So last but not least, I'd like to thank the following funding bodies and collaborators, and thank you for your time and attention. And I'm happy to take any questions.
Thank you, Dr. Zhong, for your fantastic and insightful talk. I'm sure everybody felt very impressed by your achievement and also your willingness to share with the scientific community to facilitate drug discovery, biomarker discovery, as well as basic research. And I'm sure as a comprehensive resource and a tool for studying, it's not only gonna be useful for proteomic research, but also many areas of research, and hopefully we can achieve the goals of personalized medicine. We now can go into our panel discussion and see if our other experts would have some questions or some interesting points that they would like to share and add into this great topic.
Sure. Tina, you want to start?
Yeah, let me start. I have a lot of questions. I just worry about too many questions.
That's great. We have time. We have 32 minutes.
So you have a very interesting claim. You say PPI is better captured by proteomics. So what's the evidence for this? Because now a lot of people are interested in PPI, and they are indeed very important for both biology and also for biomarker for drug discovery. So
let me share the screen again.
You only profile the whole proteome. Then how do you conclude the PPI is better presented by the whole proteome data instead of doing a lot of
it's a very good question. And to be honest, the paper was submitted as a resource because of this kind of validation, right? We did, certain analysis, but only on the data we have. Sorry, I think it's the part of here that we claim the PPI is and it's better than the others. So can you see my slide?
Yes, yes.
My thing is here, right?
So in this slide, how did you make this conclusion, point 3?
Just let me think back to it to find out whether these figures support that. Because this is from the paper, maybe it's in the supplementary. Yeah, I think these three figures do not, are not here. That thing maybe it's in the supplementary figures. Sorry for that.
Okay. I have another question. So one of your slides showed only about 1,500 proteins, which construct a subset of the network. You claim that it is sufficient to predict drug sensitivity or whatever phenotype. So are these 1,500 the most abundant 1,500, or they are specific group of proteins which may have some abundance protein and some low abundance proteins?
Okay, very, very good questions. So actually, figure C answers your question, and also the reason why we did figure B and figure C is exactly I wanted to know what you just asked. Here, right, it's a pure computational measurement. So we reached the mean Pearson's correlation R 0.433 if we have 1,500 proteins, and the maximum we can reach is 0.49. So this is the claim of 88%. But people also ask me, what are these 1,500 proteins? But in order to, instead of analyzing each of the 1,500, which is out of scope of this publication, what we did is to categorize the proteins in category A, B, and C. And we did find out proteins that have high abundance or that's annotated in the protein-protein interaction network actually have a better predictive power. And one reason is that people have the abundant proteins are these proteins people actually have analyzed in their in all the studies. So that's the only explanation. Or maybe protein are really highly connected. That's why these will explain the biology.
So you mean the more abundant protein is more highly connected?
Also, they are very correlated. We didn't do this. So you can see here the degree of connectivity, right? So category A, those proteins have great sample support, and exactly they have more connectivity in terms of the degrees. So you can calculate the edges to the node.
Yeah. So for group category A, B, C, do you categorize based on the protein abundance?
No, but we actually have figures. I asked the question, it's not here, but I can tell you they are.
To my understanding,
category A, not only they have great sample support, but also they are highly statistically significant, highly abundant proteins than category B and category C.
Because I know there are many transcriptional factors. They are regulating hundreds of genes, and they are relatively low abundance, like a TP53, and a lot of high protein kinases. They are relatively low abundance, but they are highly connected to many other proteins. Or they belong to category C in your plots.
Yeah, that might be one of the reasons.
Yeah,
I
know
suppose.
In many manuscripts or in the view of many reviewers, the high abundance proteins may not be so important. And a lot of people want to search for the low abundance ones. But here your analysis shows the high abundance proteins are also highly connected. They represent some informative network, which can be used to robustly predict cellular phenotype. This is
Mhm.
something very interesting.
But do you think the sample support is more important than protein abundance? Because here I remember or I recall the reason why in the end we defined category ABC in terms of sample support is that, you know, it correlates with the degree of connectivity and protein-protein interaction networks. It might be, so what do you think? Maybe it's better than abundant proteins? If the sample support, for example, this protein is expressed across 90% of samples, rather than this, you know, protein is highly abundant in a few samples.
So because what you analyzed are all cell lines, although they from more than 40 different tissue types. So
Yes.
cell lines have some commonly expressed proteins. If you see some protein which are expressed or detected in let's say over 90% of the samples, it's very likely it will be a relatively high abundance ones. So it means they like acting like
housekeeping.
Yeah, housekeeping.
It's a housekeeping
Blue,
proteins,
blue
right?
ones,
Yeah,
So,
that is also true.
So sorry if I may interrupt. Is it possible that when you're looking at the high abundance protein, there's a higher possibility this protein have been, so like a protein-protein interaction study like pull down or whatever you name it has been performed on this protein, and that's why when you look at database like Quorum, BioGrid, you are more likely to find this. Yeah. So that is not because highly abundant protein tend to have more network, but highly abundant protein tend to be more experimented.
Right. Experimented. So we actually wrote that in the discussion because one of the reviewers were asking the same question, right? And our hypothesis is exactly these proteins are better or you know more studied than the others. That's why they're in the network.
And my second question is, if you look at the protein abundance and also co-regulation, right? So you mean to say that you're looking at co-regulation as indirect inference for protein-protein interaction, right? Because so I have studied with this Win signaling, like this freezer complex. Seems like most of the receptor component, they are regulated in the same way. So you have one subunit of the receptor. So when you look at their co-regulation or co-expression, they tend to show the same pattern. So, but it cannot be said that all co-expressed protein should come from the I mean should form protein interaction. It could be just the same transcription factor is binding to the promoter or whatever, or not multiple related genes. So, but it's very interesting study.
So Tianan, I can go back to your question, which I couldn't answer. Now I find out which figure is showing this. So can you guys see my screen and figure G?
Yeah.
Can you see the, So I think this is analysis, right? Here you can see we have proteomics, transcriptomics, CRISPR, and merged, and using 4 database here. And we calculate the area under the curve, and we show that proteome, you know, it's better than the others. Of course, the best is a merged one. So I think this is analysis we performed and then claimed a PPI, sorry, the PPIs are better captured by proteomics.
You mean you use the whole proteome data for co-expression analysis?
I think so.
I see. The column is protein complexes.
Yeah.
A string has more protein.
So we use 3 or 4 of them: Coral, BioGrid, string, and HuB.
I see. This makes sense. Thank you.
Right, and then consistently, sorry, I didn't see this. I'm a little bit late.
There's another question from the audience online. There, can you tell me, tell them? So this haha. So he or she asked, can you tell us more about your batch design? In your batch design, did you mix cancer cells from different origin, from different tissue types, or did you run the same? type.
A little bit complicated because when Sanger team were doing their biological preparation of the cell lines, it's already totally randomized. And then they send, here you can see, right, that's the batch design we have. And you can see the first batch they sent to us is in 2018, and the last one is 2020. And we randomized it again when we do the sample preparation. And then we randomize it again when we put them into the one of the 6 mass spectrometers. So surprisingly, when we plot the, sorry, let me show you. When we plot the UMAP without normalization, we couldn't see any batch effect already. So it's, I'm not sure why is that, and because we see a lot of batch effect when we are dealing with tissue samples, but it is it may be because of several randomization at different stage.
Hey,
but we
Tim,
still used an internal normalization from Dyne.
Yeah.
And the batch design is very simple, total randomization, I think, for this project.
Mm.
That's the
Yeah.
only project in ProCan we have total randomization due
see.
to technical reasons.
Mm. So can you go to the UMAP?
Sure.
So here, all the cell lines, the cancer cell lines, do you have controls? Let's say, I don't know which control you use, 293 or HeLa cell as a control that you run in each batch and see.
We have HEC. We have HEC yeah, we have HEC samples.
Yeah.
We have HEC samples.
If you put a HEC samples in this plot, will they cluster together? I think they will cluster
Yes.
together. But
Yeah,
for the different batches, do you see batch effect?
Yeah, they all cluster together. Yeah, they do. And to be honest, we put HEC to all the projects in ProCan. We always see they cluster together very well. So that's the internal control we have. I think you know that too, right? From the very beginning.
Yeah, sure.
Great.
Yeah, and sorry to interrupt. And for the batch effect, just curious, and what's the most like the serious batch effect coming from, like the digestion batch or the instrument batch or the sample preparation batch or the like the technical batches? So do you have to weight them?
Don't really have, as I said, it's surprising. But if you want to ask what is the high, the biggest batch, I think is the machine. It's not the temporal ones. So every time when we receive one, right, we put into different machines. So here's the machine. So for example, the first P02 is batch 1, right, and we put in machine 6 and machine 4.
Yeah, and I also maybe have some technical question about the DIA data analysis. So, so you mentioned you use Diane to analyze this large cohort, and yeah, I think many people like it. And I'm just curious, did you also try to use open SpectraNote or open source to analyze them?
Yeah, it's a great question. We did use open source in the early days. And with open source, we only got 3,700 proteins quantified. And we were very worried about the suitability of publication of anywhere. So somehow internally, we decided, also I lobbied a lot to use Dyne. Fortunately, exact the time when we had the data, the Dyne came out, right? And we actually asked the developer to help us, because at the time the version is not very stable, so he helped us a lot, and we finally achieved and find out that Dyne is really much better than open source for this data set. I don't want to overclaim. And we didn't try others. We only tried open source and Dyne.
Yeah, okay. And yeah, to follow this question, usually I also think the library, the spectral library, plays also a very important role for DIA data analysis. And I noticed that in this study, you actually use like in silico spectral library, and I'm just wondering why, and why not you use like a more like typical library which was generated by DDA files. So did you also try to generate a DDA library?
Yeah, actually we did. Because with open source we need it, right? So we did DDA, and it didn't work very well. Maybe because of the heterogeneity of the data set, and we find out that the DINE library is good enough.
Okay.
Yeah.
Yeah, but somehow DIAN can also use a DDA library. Did you try?
Yeah, it can. I think we tried, and finally we decided to use the in silico one, I suppose. Yeah, I'm not so sure, but if you read this in the manuscript and the method part, then it must be true then.
So with this in silico library means you extract library from the DIA files. You have 6,000 DIA files. Well, that's pretty comprehensive.
If I recall correctly, maybe they did a sampling of it, not all the 7,000 stuff. So if you see the timer, right, we really started to do DDA and open source back in 2018 or 2019, and then I couldn't remember, the DINE is 2020. When was DINE online? The code I can't remember, and then we really switched. It's a huge, move, and I think it paid off. And we were really happy, the existence of DINE.
Okay, so Tim, I have another question. So you in one slide you showed you have 132 drugs which are connected to protein one step away with targets. I think this is a very interesting claim and also a very interesting finding. Can we go to that slide?
Yeah, this is the one I think.
Yeah, exactly. So
yeah, so these are color coded with here is the PPI distance, right? You have blue, gray, light gray, and orange. So
T means drug targets, is it?
Yeah, T denotes the associated protein is either a canonical target of the drug or CRISPR-Cas9. So we have here one is for drug protein and the other one is for CRISPR-Cas9.
So one means is one step away from the target.
Yeah.
Two means two. Okay.
Yeah.
How about the blue ones? The blue ones.
So the blue ones are predicted. So blue ones are actually significant ones because it's, if you see this one, but they are not with the target because target is coded. It's color coded with orange, as far as I understand. So they are not overlapping, right?
So my question is, how do you explain this? So some of these drugs, they are associated with not the direct target. Maybe you didn't detect the drug target, but is the protein either upstream or downstream or interacting with this drug target? So why this happened?
Oh no. This future work has to be done by this. It's online. Everyone is welcome to find this out.
Yeah, I think I have some explanation for you. So probably if you have a drug which is a kinase inhibitor, then it may not affect the expression level of this particular kinase, but it will affect the maybe downstream proteins, upstream of this kinase, because they are regulating or regulated by this phosphorylation of this kinase. I mean, I think in clinic this has a very important role. For example, the PD-1 targeted therapy, they are not very effective in breast cancer, but breast cancer have a relatively high level of PD-1 and PD-L1. People don't know why this happens. Probably because the proteins interacting with PD-1 and PD-L1, they are the key determining whether the the patient will be sensitive to PD-1 based therapy.
So what analysis do you suggest we can do in future? Or do we need more data to support the analysis or any conclusion of it?
Yeah, I think it'd be very interesting if you have the cells, culture them, and treat with a drug. And after treatment of the drug, you measure whether the target or the proteins with 1 or 2 or 3 steps away from the target, whether they respond to the drug perturbation. If they are really responding to the drug perturbation, means they have a causal connection. It's not only a, so now you only have the whole proteome. So you cannot tell whether this, you can only generate a hypothesis. They probably associate with the proteins surrounding the target.
Yeah, we do think this paper is more a resource than a traditional article, and yeah, it needs a lot of validation, biological validation too, for the claims we made. We actually, through the various review circles, we have tuned down a lot of our claims. Yeah, and I understand that.
I was wondering for the drug intellect in drug protein interaction. So if you have a lower p-value or you have a higher negative log 10 of your p-value, and if you look at the predicted protein association, right, they are not hitting the target. But then is it possible that the I think for structural biology, people are using docking, for example, to look at if the small molecule actually can bind to other proteins, like off target, right, or not. So
yeah,
that is used for drug repurposing, right? So if a drug cannot bind to its intended target, but sometimes it can hit secondary or tertiary target, which have the similar local substructure that and the drug binding pocket is similar to the target.
Yeah, true.
So my another question is for your in silico database, in silico spectral library that you generated, you would consider proteogenomic sort of like SNPs or maybe indels, whatever that is available in that one of some of these cell lines. And that is first question. And my second question would be, of course, then currently the area like this small open reading frame, this microprotein are very hot. And in fact, in 2000 and in July this year, there's a paper published. They actually consolidate like many different data sets from ribosome profiling, and with very stringent parameter, they can downsize the list of this microprotein to 7,000. So it'll be interesting also to incorporate this microprotein, in the spectra, in silico spectral library, where we want to see, is it possible that to locate this microprotein, because it has been shown that some of these microprotein are really playing in very important role in the sort of the different hallmarks of cancer, right? So that would be something interesting apart from the known universe of the proteome, right?
Yeah. Yeah, so the simple answers to your questions are, well, no, we didn't do that. And I'm not sure whether we want to do this soon, because we have some other stuff, priorities about this data set. Because one of the senior authors is an expert in telomerase, right? So this will be dealing with that. Another one is we have a lot of tissue samples. We want to understand the relationship between this data set with our pan-cancer tissue-based data sets. So these are the two future works, but all other suggestions or collaborations we are happy to take, or everybody can just remind this data set and to them what they think it's their area of expertise.
But I find really impressive that you finished like 10,000 runs in like a span of 1.5 years, and at the same time from the protein abundance, a very traditional proteome profiling results. I mean, the data you can extend with bioinformatic, CRISPR drug binding and different things. So we do not have to go to like for example, co-IP experiment. You know, we could have just done this and then get an initial hit, and we validate and see such interaction actually happen.
Yeah, I could review a little bit more in this context is that we reran the whole stuff again, but this time with a new sample preparation method. So we expect that our protein numbers might hit 10,000 mark, hopefully. The only downside is instead of 6 replicates, the rerun is only triplicates, but still we expect the result might be even better, but it's still running now. That's only I can review at this moment. So we hope everyone can use this data set, because when we do analysis, all the problems we normally facing is that there's no validation data set. People are looking for them, especially in large scale proteomics, right? And we hope this could be a validation data set for many researchers who are interested in that.
Hi, Jing, have you done analysis to see what are the tissue specific protein or pathways? Where you have so many different tissue types, but I noticed some of the tissue types you have only 1 or 2 lines, but some of them you have quite a lot of lines. Just now, another audience, online audience, they ask, seems the lung cancer is different, very different from the others. Yeah, the lung cancer is very different from the others. But for the other cell lines, well, you have a lot of hematopoietic and the lymphoid cell lines. Cell lines. So have you analyzed their difference?
No, we didn't for this project. But what's nice to see is that these blood and lymphoid cell lines are always close together, and this is also, I mean, it also aligns with the results from CCLE datasets. When you have a look, they only have 300 something cell lines. They also have blood and lymphoid. They also see blood and lymphoid cells have their own and distinctive clusters. For that reason, we haven't analyzed. And yeah, but that said, if the second project, third project would be to understand the relationship between the cell lines with tissue samples. And we know the ProCan, it's not really cell lines, right? Are tissue samples. So we have lung cancer, and we have a pan-cancer study with a variety, maybe 1,000 samples of different cancers. We also have lung cancer as a single cohort. So internally, we would understand whether we can train a model from the cell lines and test the model on the tissue samples, and whether to see whether it can predict, right? And then find out what are these proteins, what are the differences in terms of their network and their abundance. So these are all the questions we actually are solving at the moment.
Then do you have the doubling time data for each of the cell lines?
What do you mean sampling?
So, different cancer cells, they have different aggressiveness. Some cancer cells grow very fast, some maybe slower. And this is, so then we can, if you have the data for the doubling time, it means how long it takes for the cells to double in number. Then we can compare those rapidly growing cells to those slowly growing cells. Probably there are some pathways driving the cell growth and make some cells grow faster than the others.
That's very good. I will have to find out whether we have this metadata, or maybe this already we have it. Then we can investigate. It's a very nice one. Thank you for the suggestion.
So Liu Jia, do you have a question? Just what you want to say.
I have a question about its clinical application. Can we use these results into clinical samples directory? For example, if I have performed proteome profiling of carcinoma samples from a cohort patients, how can we use these results to help stratify cancer subtypes with different job response? Could you please give us some suggestion about such as data processing, or could you give us more application scenarios? Thank you.
Thanks for the question. I think it's still early days that we could, a lot of people do not believe cell lines. Cell line, for basic biologists, they are very good models, but for clinicians, they're really far away, right? They're not really tissue samples. That's why I mentioned the next big project for us is to get a view about how cell lines are related to tissue samples, at least at the proteomic level. And then we can, because cell lines, it's according to Roger or it's my boss who's a biologist, is a more pure population of a cancer. So it's easy to study if we can establish relationship with tissue samples. So we can do a lot of things, but there's a trajectory. What you are talking about is, can we already use cell lines? I think it's no. From cell lines, you have PDX, you have organoids, right? All these kind of things. So we're still in early days, I think. But once we establish the relationship between them, maybe proteomic measurement is stronger than genomic or any others, then we are really happy. And then everybody will think the proteome analysis will make some difference in clinics. So recently we published a paper, it's a viewpoint about what is a pharmacological, proteome-based pharmacological screens, right? We plot a figure really showing cell lines, and then organoids, PDX, and then tissue samples.
So can you send me the paper? I'm very interested in this paper.
Oh, you are the editor. You invited me to write it. So as an editor, I suppose you have no, it's published. It's online.
It's
online now.
Okay.
Yeah, it's online. It's
which journal? Which journal is it?
In proteomics.
Proteomic. Okay.
Proteomics.
You even forgot that you are the editor.
He's
it's independently reviewed, so
yeah.
I'm not aware of this, but
yeah.
I'm very keen to know this. But now a lot of people are interested in drug discovery and how multi-omics, particularly proteomics data, can help in drug selection, drug discovery. So just now you also mentioned some CRISPR data. So the CRISPR data are these online data, existing data you just use, or you perform experiments to acquire this data?
And I think I mentioned in one of my slides that the Sanger team has expanded their drug response data. So some of them are already there in the in a cosmic, or they call it cell. It's very confusing how they name. Even people within Sanger Institute, they have actually different databases. They have some politics I am not aware. But essentially, in this paper, we not only provide the proteomic measurement of these cell lines, also we provide more drug data. Also, I think CRISPR, some of them are new. So we are still collaborating with Sanger. We have new ideas, which I cannot review here, but it's a continuous collaboration with them. Because with these results, they are happy with us, we are happy with them. So,
cool.
We'll be continuing.
Yeah. Sorry, ProCan is pretty much focused on proteomics, especially mass spectrometry based proteomics, right? So if
actually
not, you also do
so we actually do multi-omics. So we do not have samples because we are a research institute, right? So we get samples from collaborators worldwide, as Tianan did it's the same from Germany, from UK, from everywhere. And we only actually accepting samples which already have genomic or transcriptomic or some other data types. And then we just add proteomic measurement on top of it. So essentially we do integrative multi-omic analysis. If you see all these big papers this time published, either it's COVID or you have to be integrative machine learning, AI, or, right? Or pan cancer. Otherwise, it's so difficult now these days to get published.
So Hai-Ching, did you also try to look at proteoform, for example, isoforms or PTMs into this large data set? Not yet, or not interested?
No, not yet. Not yet is the first answer, because these are recurrent questions. So every time I present something, people ask, have you done this? Have you done that? I think the protein is more clinical relevant. So we want to do something that can be translated in clinics, meaning we need some results within 24 hours or within 72 hours. So doing all the other stuff might be contradictory, or it doesn't match to the clinical goal. But maybe it's important. But now we haven't tried the protein abundance. We just want to use protein abundance with other data to inform clinical decision. If that's not possible, we can always add more. If the technology is after 5 years will be different, then we can always do that. But for the moment, our official answer is we can do, but we haven't.
So you mentioned you collect data samples which have multi-omic other omics data than you acquire proteomics. Then in your experience, which omics is better for, let's say, predicting phenotypes, for drug sensitivity prediction or diagnostic markers?
Exactly.
My answer will be biased, of course, proteomics, right?
Because my question is also that if transcriptomic RNA-seq has been very sensitive, and people can do like single cell transcriptomic quite relatively easily compared to single cell proteomics, and how would the current generation of transcriptomic RNA-seq data compare to proteomics? Is there any difference that proteomics can make in this respect that we can find? For example,
when we're thinking about the correlation between RNA and protein data, right? This is the question, also a recurrent question, and Yan Shen published a review paper about that. So he wrote in his paper that he did a survey. It's about 0.4 to 0.6 is the RNA protein correlation, and we now have analyzed more than 20 or 30 projects in protein, including this one. And I can tell you that it's really the range. This is actually a good thing, right? Because some of them you can say, okay, they are the same, but some of them maybe open new avenues, something only happening in the proteomic data set, which is why we are working on these data sets, and I hope they are useful in the end.
At least in your case, you have shown that with respect to PPI, proteomics is give a better estimate, right?
But is that a cheating? Because the PPI is called protein-protein interactions. Then people might expect the protein can predict PPI better than the others. I'm not sure. I'm not. Tiana, do you think so? Just by the naming of the network.
Yeah, I think we need more experiments. That is beyond the scope of this study. That's a very, very important issue, but I think we need more evidence to show that.
So a little bit off topic here. I have a question for Dr. Shah. Sorry. So you mentioned about proteoform measurement with the DIA, right? So I'm just wondering, because I'm not following up to the latest, that happenings in DIA, how easy is it to generate an in silico spectral library for this proteoform? For example, like phosphorylation, that would be so complicated, isn't it?
Yeah, I think you mentioned the right question. I think like for the library part and the phosphorylation phosphor library, it's not easy, but and the nice part of DIA analysis or DIA measurement is it captures all the signals, including the phosphopeptides. So the phosphopeptides, yeah, there maybe it's not that abundant, and because also there's no, I guess, not phosphor enriched like some preparation step, so that, but it provides like a nature biased way to look at all the proteoform of your sample, including like phosphorylation, oxidation, and the methylation, and all the other stuff. But for the PTMs for the library of PTM peptides, I think it's not very easy to predict. But like the Diane, the nice thing is it can generate in silico library directly from the DIA data.
We lost him.
We lost you.
Very critical, very important issue. If we can analyze PTM from the, I believe so, if there's a reliable algorithm to dig out a PTM from this data set, that'll be very powerful.
Yeah.
Hey, Wenguan, we lost you for maybe 30 seconds.
I'm sorry. So from which part you lost the connection? Yeah, maybe I can repeat. So I think the phosphopeptides, it's there. It's the nice thing of DIA measurement. Yes, it records all the signals, including peptides with different proteoforms. But one not very perfect part is we do not do a specific protocol to enrich the peptides. Yeah. And for the library part, one nice thing for the Diane is that it generates in silico library directly from DIA files, so that it overcomes the problem that we and the proteoform peptides is very difficult to predict. But we can use generate the pseudo spectrum from the DIA files, which is exactly what Diane is doing.
Yeah, thank you.
Thank you. So, Liu Jia, do you have another question? Any question? We should encourage the students to ask more questions.
He's smiling.
Okay, it's fine. I think we already spent 1 hour and 12 minutes. I think it's time to conclude. Thanks a lot, Jing, for this very insightful talk. It's not easy to generate such a huge amount of data with over 6,000 DIA files in a single project for so many cell lines, close to 100 cancer cell lines. I can see from your timeline over 2 years of data acquisition. I think
only for data acquisition.
Yeah, even more time for the data analysis. I notice this is a very special cancer cell paper. You have no mechanistical validation, purely data analysis. But I notice you have developed a few algorithms to find the biomarkers, to the networks, and you have very interesting insights concerning drug discovery, diagnosis, and I think this is very interesting resource. I'm also very keen to download data and analyze them together with our tissue data. We have some tissue proteome data also from different types. It will be interesting to see whether how different is the pure cell lines, in vitro cell lines compared to the tumors. And thanks a lot for Tech and for Wenguan and for Liu Jia for joining this section. And we have very good discussion. And thanks for the audience online. So Bing, do you want to say a few more words?
I think it was already been a great wrap up. But yeah, it's been an honor to be among such an amazing lineup of accomplished scientists today. And thank you for being here. Hopefully our ongoing collaborations will lead to more fruitful discoveries that lead to more helpful, fruitful discoveries that can help the scientific community. Thank you so much. Yeah.
So much.
Thank you. Bye-bye.
Bye.
Thank
Bye.
you. Thank you. Thank you, Ching. Yeah, I have
Bye.
to go home and spend some time to try to read more and understand about the data analysis part.
Sure.
Okay.
Thanks, everyone.
Very impressive. Yeah,
Thank
thank you.
you.
Bye-bye.
Thank you, everyone. Thank you.
Have a good night. Bye-bye.
Bye.